Hi, my name is Jace Flansburg, and this is my project. It's called the Inhibition Potencies of Naturally Derived versus Synthetically Manufactured Antibiotics, Effects on the Gut Microbiome. There's been an area of active research about the effects of antibiotics on the gut microbiome, like the long and short-term effects, and what type of antibiotics increase or decrease different types of bacteria. According to the National Health Service, 1 out of 10 people experience side effects that harm the digestive system after taking antibiotics. Therefore, if you can find a natural remedy that can do the same jobs as antibiotics, then they can help us stop the harming of the gut microbiome. Throughout my experiment, I plan to answer the question, how do alternative remedies affect the gut microbiome compared to standard antibiotics? This research is important because we can learn the least damaging type of antibiotics or natural remedies to the gut microbiome, but the most damaging to bacteria that causes infection. A microbiome is a community of microorganisms such as bacteria, fungi, and viruses that inhabit a particular environment. The human body needs the gut microbiome because it helps us digest foods that our stomach and intestines cannot. The microbiome helps defend the body from harmful microorganisms, helps produce some of our vitamins, and helps tell our immune system what is good and what's bad. I'm using Antibacterium aerogenes because it's a bacteria that is commonly found in the gut microbiome, but that it causes gastrointestinal tract infections. I'm using E. coli because it is found in the gut microbiome, and it has an important role in keeping your gut microbiome healthy. The two key features that lead to the differing visualization properties of gram-positive and gram-negative species are the thickness of the peptidoglycan layer and the presence or absence of the outer lipid membrane. My research question is, how do alternative remedies affect the gut microbiome compared to standard antibiotics? My hypothesis is, if I introduce antibiotics to bacteria, they'll have a larger zone of inhibition than alternative remedies because antibiotics are made to kill bacteria. My independent variable is the type of treatment. My dependent variable is the zone of inhibition. My controls are the time in the incubator, the temperature in the incubator, the type of antibiotics and natural remedies, the type of bacteria, and the size of the petri dishes. For my experiment, I started off creating a mix of agar and bacterial broth for each of the bacteria I'm testing. I poured the mixture into 5 petri dishes for each type of bacteria and let the agar cool. I then prepared 10 honey discs and 10 garlic discs by placing them in a petri dish and adding 1 drop of honey or garlic juice to each disc and let them sit until the substance was absorbed into the discs. I labeled the E. coli dishes with EC or the Antibacterium aerogenes dishes with EA. Using sterile forceps each time and using the sterile technique, I placed 1 blank control disc and a single disc of each antibiotic type and natural remedy to each of the 10 petri dishes. I made sure the discs were equally spaced and labeled. I let the dishes sit upside down in a 37 degrees Celsius incubator for 2 days. I then measured the zone of inhibition using the metric side of a ruler. I measured the diameter of inhibition for each disc and recorded the results in my data tables. I disposed of the dishes by squirting bleach into each dish, sealing with tape and placing it in the school's biological waste bin. The antibiotics I used for my tests were ampicillin, penicillin, and gentamicin. The natural remedies I used for my tests were honey and garlic. In reviewing my results, gentamicin was observed to kill the most Antibacterium aerogenes and the least E. coli. The average zone of inhibition was 22 millimeters for Antibacterium aerogenes and 17.6 for E. coli. Garlic, honey, and ampicillin were observed to kill more E. coli than Antibacterium aerogenes. Garlic had a zone of inhibition average of 12 millimeters for Antibacterium aerogenes and 18.2 millimeters for E. coli. Ampicillin had a zone of inhibition average of 10 millimeters for Antibacterium aerogenes and 29.2 millimeters for E. coli. And honey had a zone of inhibition average of 1.8 millimeters for Antibacterium aerogenes and 29.2 millimeters for E. coli. The honey was also observed to kill the least Antibacterium aerogenes and the most E. coli. Penicillin was observed to kill none of the Antibacterium aerogenes or E. coli. To determine if the difference in zone of inhibition diameters for each antibiotic or natural remedy between E. coli and Enterobacter aerogenes is likely to be significant, and a ANOVA test was run. I'm using it in an ANOVA test, or analysis of variance test, because it will determine whether there are any statistically significant differences between the means of different groups. After I run the ANOVA test, I got a p-value of less than 0.01 for the Enterobacter aerogenes, and a p-value of 0 for E. coli. A p-value less than 0.01 is considered statistically significant. Because the p-value is so low, it's very likely that the differences in the zone of inhibition diameters for each antibiotic or natural remedy between E. coli and Enterobacter aerogenes are not due to random chance. I also did standard deviation for all of my data sets. And the low standard deviation, which is a standard deviation that is less than 2, tells you that your data is closer to the mean. Low standard deviation also increases the likelihood that the independent variable is causing the change in the dependent variable. A high standard deviation, which is a standard deviation that is more than 2, tells you that your data is spread out. A high standard deviation also means that additional factors other than the independent variable could be affecting the dependent variable. The data collected in this experiment supports the initial hypothesis that if I introduce antibiotics to bacteria, then it will have a larger zone of inhibition than alternative remedies because antibiotics are made to kill bacteria. Gentamicin was most effective against Enterobacter aerogenes, but it was also effective against E. coli because it's a gram-negative antibiotic which works best against gram-negative bacteria. Gentamicin is an aminoglycoside class antibiotic, which inhibits the synthesis of proteins in bacteria, eventually leading to cell death. Garlic was most effective against E. coli, but it was also effective against Enterobacter aerogenes because it's a natural gram-positive and negative antibiotic, because it has a compound called allicin, which is a powerful natural antibiotic, about 1/50th as powerful as penicillin. Ampicillin was most effective against E. coli, but it was also effective against Enterobacter aerogenes because it's a broad-spectrum antibiotic which works against both types of bacteria. Ampicillin is an aminopenicillin class antibiotic, which works by inhibiting bacterial cell wall synthesis. They're chemically similar to penicillin, but have a broader spectrum of activity than penicillin. Honey was most effective against E. coli, but was also partly effective against Enterobacter aerogenes, because the honey contains hydrogen peroxide, which may account for some of this antibacterial properties. It also has a high sugar content, which can help stop the growth of certain bacteria. Additionally, honey has a low pH. This works to pull moisture away from the bacteria, causing the bacteria to dry up. Penicillin was not effective on either of the bacteria because a gram-positive antibiotic trying to kill a gram-negative bacteria. The data collected in this study was limited by the amount of materials, so I could not do more than 5 tests, which is something I would change in the future to something maybe like 10 tests. Also, I was limited on the types of bacteria I could test. With this research, we can learn what types of antibiotics or natural remedies will kill the most bacteria that is causing infection in the gut microbiome, but keep the most bacteria that helps keep our body healthy. Future research could investigate the best type of antibiotics to protect the gut microbiome, or the best probiotics to take while taking antibiotics to keep a balanced microbiome. Research could also be conducted to find how antibiotics affect other microbiomes, or how to keep these microbiomes healthy while taking antibiotics. This research would be particularly important for us to find out how to keep our bacteria balanced while taking antibiotics. I got help from numerous science teachers at my school that helped me plan and execute my experiment.